A safe and a blessed morning to everyone, or might be in your case, good afternoon and good evening. I am Royet Puyo, and I will be delivering this week's topic discussion on the ABO blood group system. And since this is your immunohematology lecture, the discussions will be a little less on the laboratory practical aspect, but a little more of your theoretical perspectives of blood banking. So, in short, we are expecting to have a wider range of topics and, well, of course, possible clinical correlations that may as well facilitate the uptake of the necessary information that we all need to learn from. And one more thing, since we will not be giving you your lecture handouts apart from the PowerPoint in PDF form, I am enticing everybody to take down all the important information that are verbally delivered, most especially the several board exam pointers that I will soon be emphasizing throughout the discussion. So we have here are the learning objectives like we always do have, and since this is part of an outcome-based learning. So okay, we have listed here only a few, but it's generalized learning goals that everyone must or is expected to accomplish after watching this pre-recorded lecture presentation. So number one, differentiate ABO genes as to their functions and involvement in the formation of the structural differences of your ABO antigens. We have number two, describe, enumerate, and differentiate A and B subgroups. Number three, discuss Landsteiner's rule and describe the relationship of your reverse typing and forward typing results. Number four, learn ABO typing results interpretations and correlations. Number five, learn, describe, and differentiate the genetic basis of your H deficient phenotypes. And lastly, we have your number six, describe, identify, and interpret ABO discrepancy and be able to give their corresponding resolutions. So, beginning with a brief overview of the historical perspective in the ABO blood group system. You know, class, it is always imperative to at least get acquainted with the milestone, most especially in the scientific evidence of numerous discoveries, including the contributors that comes with each discovery. So Landsteiner, De Castello, and Sturley are known as the three blood bank pioneers that were recorded in our blood bank history. The ABO blood group system was actually discovered and was first described in 1900s by Carl Landsteiner and was officially reported by the year 1901. So according to our references, Landsteiner extracted blood from himself and alongside with five other constituents. He separated the sample serum from the red cells and mixed these red cells and serum obtained from the different constituent samples. Landsteiner was actually able to classify three different patterns of reactivity and was initially termed as A, B, and C, and in the latter years was reclassified as your A, B, and O respectively. And this actually has made him the very first human to perform the ABO blood grouping. So while in 1902, Von de Castello and Adriano Sturley both are actually colleagues of Landsteiner, and they discovered the blood group AB. And I believe, if not all, almost every one of us knows someone who is blood type AB, and this is the rarest of the common ABO blood group. Okay, now class. In board exams, if you were asked about what specific year did Landsteiner discover the ABO blood group system, and both 1900s and 1901 are in two separate choices, always chose the letter corresponding to the year 1900s. So as you can notice or observe for those who have been reading Denis Harmening, 1901 was indicated as the year of discovery of your ABO blood group system, but from other references like Henry's and multiple others, the details goes the same, like 1900s for the discovery and 1901 for the official reporting of the ABO blood group system. In your human blood bank, we actually have multiple blood group systems, which of course includes your ABO blood group system. Your ABO has the ISBT number 001, which is the nomenclature given by the ISBT, or your International Society for Blood Transfusion. We actually have more than 700 blood cell antigens that was grouped and organized by this association, and they grouped this into only 30 blood group systems. But among these 30 blood group systems, your ABO is the most important and most clinically significant of all the blood group systems in the transfusion practice. And why exactly? It is because this is the only blood group system in which antibodies are consistently and predictably present in the serum of people who lacks the corresponding antigen. And the blood group of a person is actually based on what antigen is present on his red blood cells. So for example, if a person has antigen A on its RBCs, it means that this person is blood group A. If one individual has antigen B on his RBCs, then this individual is blood group B, and so on. And we will try to broaden these concepts as we proceed with the succeeding slides. The production of your red blood cell antigens are genetically modulated and are actually the results from the interaction of genes at three separate but related loci. You have your number one ABO genes, number two H genes, and number three SE genes. Your ABO genes are located at the long arm of your chromosome 9. H genes are also known as FUT1 gene or fucosyltransferase 1 gene is located on the chromosome 19 and that are needed for the formation and development of the structural composition of your ABO antigens that are present on your erythrocyte membranes, meaning to say they are present on your RBC membranes. Next, SA genes are also located at the chromosome 19. They are needed for the formation of ABO antigens on soluble substances or your body secretions or body fluids. Okay, this is a board exam pointer. Take note that the difference between H and SA genes are not on the chromosome location. When we say H gene, they are genes needed for the formation of ABO antigens on the red blood cell surface, while S genes are needed to form ABO antigens on your body secretions. Next, we have the inheritance and genetic makeup. So in 1924, a scientist in the name of Bernstein postulated a theory of the inheritance of your ABO blood groups. He stated that your ABO blood group has a unique genetic makeup and that it follows the Mendelian genetic laws. Okay, this is a review and one formal correlation of your basic human biology and particularly your cytogenetics. We all know that the basic principle of your Mendelian law explains that the hereditary units occurs in pairs and that these units will undergo a separation process during your gamete formation so that the resulting progeny will acquire one allele from each of the pairs, meaning to say one allele from each of the parents, where both of the acquired alleles are genetically expressed. And that Bernstein explains that your Mendelian genetic law applies to the inheritance of your ABO blood groups via codominance or in a codominant expression. And the designations of A and B refers to phenotypes, whereas AA, BO, OO, and so on denotes for genotypes. However, your O gene here, as you can see on this present slide, this was mentioned that as opposed to your other genes that were previously discussed, it is an amorphic gene or a silent gene, meaning to say inheritance of two O genes that are non-functional or unexpressed, which also means that it does not have the capability to produce any detectable products, and that is why, and that is exactly why the blood group O phenotype or blood type O is an autosomal recessive trait. So this is a table showing your different ABO genotypes with corresponding phenotypes. As you can notice that in a group of individual, both genotype and phenotype are the same simply because, as I have mentioned earlier, that O is an amorphic and inheritance is a recessive trait, and that therefore must be in a homozygous orientation for the O gene. And so, as you can notice that there are numerics of 1 and 2 on your A's. These A1 and A2 are the main subgroups of Group A, which will be discussed or which we will be expounding in a more detailed manner as we proceed on the discussion. So now for the main concept of the formation and development of red cell antigens. So let us take the full attention at this present slide. Okay, this time we will be discussing one by one the structural composition of the oligonucleotide sugar structures or chains that forms your erythrocyte membrane antigens. Your previously discussed genes that plays an important role in the formation of your ABO antigens, namely the H genes, A genes, and B genes, excluding your O gene since it is an amorphic or a silent gene that does not produce any antigen, and at the same time your SA genes, or excluding your SA genes which are not present on your RBC membranes. So these three genes do not directly code for the production of your ABO antigens, but actually codes for the production of your enzymes or your glycosyltransferases that will facilitate the placement of different sugar molecules to the precursor substance found on your red blood cells. So, okay, a bit of review. These precursor substances are actually comprised with chains of different sugar molecules, which I am pretty sure that have been you have been encountered multiple times during your biochemistry class. They are termed as paragloboside or glycan, which is a complex, interesting structure. However, it takes another time for a more detailed explanation. So moving on. Each gene codes for a specific glycosyltransferases. Therefore, each gene will have a varying sugar molecules, and these sugar molecules are specific for each, or in other terms, the immunodominant sugar that builds the specificity for each of the antigen formed. So your each gene codes for the production of L-fucosyltransferase. So from the term itself, fucosyltransferase. It transfers the L-fucose sugar to the terminal end of your, or to the terminal galactose end of your paragloboside, and this mechanism will result to having now your L-fucose as the immunodominant sugar for your H gene. Now, the conformational change or addition of the fucose molecule to the terminal galactose, or the galactose-fucose linkage built, is now referred to as your H substance. The same concept actually goes with the other two of your genes. So the A gene codes for the production of N-acetyl-galactosamyl transferase that will transfer the N-acetylgalactosamine sugar molecule to the H substance. The immunodominant sugar formed is now your N-acetylgalactosamine. And lastly, the B gene codes for the production of the galactosyltransferase, which is responsible for the transfer of the galactose sugar molecule to the H substance, resulting now to having the galactose as the immunodominant sugar for B gene. And take note that the resulting change in the oligonucleotide chain or on the paragloboside or glycan creates now your molecular composition for the H, A, and B antigens respectively, and To know what that exactly means, let us take a look at these following slides. So we have here a nice illustration of the oligonucleotide structure of the H antigen found on your red blood cell surface. Your original precursor chain here includes the GL, glucose, GAL or GAL, galactose, and GalNAc, N-acetylgalactosamine, and the terminal galactose. Now, as I have mentioned earlier, that the H gene actually codes for the production of L-fucosyltransferase, and the action of this enzyme transfers an L-fucose sugar molecule into the terminal galactose of the precursor substance. And this whole new structure, I'm referring to the precursor with the addition of L-fucose, now builds the structure of your H antigen. As you can notice, the FUC on the gal or galactose now becomes the terminal sugar constituent of the original precursor substance. Your galactose fucose linkage is what we call now as your H substance, in which the immunodominant sugar on your H antigen is fucose, which now builds the H specificity. So, and that is your H gene. Okay, let's review. H gene codes for the production of L-fucosyltransferase that will facilitate the placement of L-fucose to the terminal galactose or of the paragloboside. The galactose-fucose linkage is referred to as your H substance, while the entire oligonucleotide chain formed is your resulting antigen structure, which is the H antigen. So now, here is a very special role of this H antigen. The H substance for from your H antigen actually serves as the precursor substance for the formation of your A and B antigen. So meaning to say, an individual must have the H antigen or must inherit the H gene in order to produce your A and B antigens. And that's exactly why it was mentioned earlier that H gene is present on 99.99% of random population. So recalling our mode of inheritance, the O gene is actually an amorphic or a silent gene that is incapable of producing antigens. Since inheritance of this gene is an autosomal recessive trait, there is no specific glycosyltransferase associated with its inheritance, and that's exactly why for blood group O individuals, they do not contain both of the antigens or your A and B antigens specifically in their erythrocyte membranes. For this blood group, it must contain a double dose of a dominant H gene alleles or a double dose of your H gene alleles with at least one dominant H allele, and it is since it is said to be an autosomal recessive trait, it must have two homozygous O genes. Okay, now since O gene is a non-functional and unexpressed gene incapable of producing a specific glycosyltransferase that will facilitate the conversion of the H antigen into other form of antigens, blood group O has the most abundant amount of unconverted H antigens. And it means to say that it also has the highest concentration of H antigens among the common ABO blood groups. So this is your A antigen oligonucleotide structure. Your A gene codes for the production of your N-acetylgalactosamine transferase that will transfer or facilitate the placement and attachment of the N-acetylgalactosamine to the H substance, which is a result from the inheritance of H gene. So now, you can obviously see or notice the main importance of the H gene for the formation and development of your A antigen. In a more technical term, the glycosyltransferase converts the H antigen into an A antigen via addition of the N-acetylgalactosamine to the H substance. The GalNAc H substance linkage is now referred to as your A substance, where your immunodominant sugar is the terminal glucose molecule and acetylgalactosamine, which also builds now your A specificity. And okay, class, as you can remember from our previous slides, it was mentioned that the blood group of an individual is determined by what antigen is present on its erythrocyte membranes. So it means by just looking at this picture, we can easily deduce that this is for blood group A or a blood type A individual. So blood group A or blood type A has a double dose of dominant H genes, or at least one dominant H allele, and a double dose of dominant A genes, or at least one A gene with a recessive O gene. So, okay, board exam pointer. Your A gene, based from scientific statistical data recorded, appears to elicit higher concentrations of the glycosyltransferase as opposed to the B gene. It means, therefore, that the inheritance of A gene may highly lead to the conversion of almost all of the H antigens on the RBCs into approximately 810,000 to 1,170,000 A antigenic sites, most particularly to an A1 adult RBCs. And this is mainly one reason as to blood group A as the second most common blood group, most common ABO blood group. Lastly, we have your oligonucleotide chain structure for the B antigen. Having the similar concept as the ones previously expounded, your B gene codes for the production of your D-galactosyltransferase, which facilitates the transfer of the D-galactose to the H substance. The addition of this sugar molecule, particularly via the action of your D-galactosyltransferase, leads to the conversion of the H substance or H antigen into a B antigen having D-galactose as the immunodominant sugar. Okay, now, as opposed to your A genes, it shows a less concentration of the transferase production or the enzyme production, converting around 610,000 to 830,000, which are present on a blood group B adult RBCs. While the mode of inheritance goes the same way as your H and A antigens, for blood group B, at least one dominant H gene allele and one dominant B gene allele genetic orientation is needed for the expression of the blood group B or blood type B. So class, in the case of blood group AB or blood type AB, both of the genes, specifically the A and B genes that codes for the production of glycosyltransferases from both of the inherited genes, which will result to the addition or transfer of N-acetylgalactosamine and D-galactose to the H substance, converting now the H antigen into an AB antigen containing both of the A and B immunodominant sugars on the erythrocyte membranes. Okay, take note that for blood group AB, even though A gene elicits a higher concentration of glycosyltransferase than B gene, but when paired with group B, as in group AB, the B gene becomes more competitive at acquiring H antigenic sites, which results in an adult AB to have approximately 600,000 A antigen sites and 720,000 B antigenic sites. Aside from your ABH genes, we also have in particular your SE genes, secretor gene, or your fucosyltransferase 2 gene. As stated previously, both of your H gene and SE gene are closely linked together and are located on your chromosome 19. And since SE genes are found on different chromosome locus from your ABO genes, it only means therefore that the production of your secretor genes or your SE genes do not affect or modify the formation of your ABH antigens on the RBC surfaces. So Okay, your SE gene also codes for the same enzyme like your H gene, which is the L-fucosyl transferase, that will also facilitate the production of H antigens on your secretions. And consequently, if a secretor individual also inherited the A gene or the B gene, it means that the H antigen on the secretion will also be converted into either B or A antigens, depending on the gene or genes inherited by the progeny from its parents. Okay, class, take note that this is a valuing board exam pointer. A person's blood group or the antigen present on that person's RBCs are also present on the secretions of that individual. From this concept, we can deduce that the ABH antigens on RBCs are predictively or are also the antigens present on your secretions. And this is exactly why long time ago in our blood bank history in the medical literature or in the past, which was recorded in our blood banks, examinations for the secretor genes were actually utilized to establish the true ABO group of an individual whose RBC antigens are poorly developed. However, this is no longer the case today due to the scientific evidences and theories that have been postulated that certain diseases or conditions may affect the production and may cause interferences to the results obtained via your secretor status examinations. Okay, this actually lead to the non-absolucy of the secretor status as definitive criterion in establishing true ABO blood group of poorly developed red cell antigens. Approximately 80% of the population are secretors, meaning to say they have inherited at least one dominant allele of the SE genes or secretor genes or your FUT2 gene, while 20% accounts for the non-secretors, which means that they fail to inherit at least one dominant secretor gene allele. So if a person or an individual is a secretor, the fluids in which A, B, and H substances can be detected through their saliva, tears, digestive juices, sweat, bile, amniotic fluid, milk, semen, and synovial fluids, which also includes your peritoneal, pericardial, and pleural fluids. So in this table shows the presence and the relative concentrations of the ABH substances or ABH antigen on secretions from secretor individuals or those who have inherited the SAG. We have here your ABO blood groups showing the corresponding ABH substances found or present on saliva, while the arrows represents the concentration of the ABH substance of each of the corresponding blood group. Okay, class, take note that these tabular representation are for those secretor individuals. So as you can notice, for a blood group O individual, it does not contain in the secretions your A and B soluble substances, but contains the H substance among the or contains the highest H substance among the other blood groups or among the other ABO blood groups, and this can be correlated with your ABH antigens present on your RBCs for blood group O. Well, as you can remember, your blood group O does not also contain the A and B antigens on their red blood cells due to its genetic orientation as an amorphic gene or a silent gene or a silenced gene, which is incapable of producing specific enzymes. But however, it contains the most abundant concentration of the H antigen. So in this table, the H substance concentration is designated by the two arrows, which means that it also has the highest amount of H substance. Moving on to your group A, Just like in the red blood cells, it contains only A antigen, which shows the same presence of A substance in the saliva and also does not contain B substance. So the same way goes with your B or your group B that shows presence of only B antigen in the RBCs and B substance in the saliva. For group AB, both of the antigens are literally or obviously present in the red blood cells. Therefore, both of the substances, particularly your A and B substance, is also present in the saliva. And this explains why it was used before to establish or confirm blood groups on individuals with weak or poor reacting red cell antigens. And as you can notice, group A, B, and AB has lesser H substance as compared to your group O. And this is because for the blood groups other than your blood group O, they code for specific glycosyl transferases that results to the conversion of the H antigens, allowing the reduction of unchanged H antigen sites. So, okay class, we actually have different definitive types of the ABO precursor chains. We have your types 1, 2, 3, and 4. However, types 1 and 3 are primarily definitive and specific for body secretions, while your types 2 and 4 are prevalent among RBC antigens or red cell antigens. We actually have 4, however, only types 1 and 2 are the most abundant precursor types and specifically definitive for secretions and red cell antigens, respectively. As for your type 1, this is actually the precursor type for your ABH soluble substances or your secretions or body fluids, while type 2 is primarily the one which is responsible for the oligonucleotide chain orientations of your ABH red cell antigen formation, and that actually leads to one more of the main characterization of these two precursor types. So as you can see here, your type 1 chain refers to a beta 1,3 linkage in wherein the number 1 carbon of the galactose is attached to the number 3 carbon of the N-acetyl galactosamine of the glycan or your precursor substance, while your type 2 chain refers to a beta 1,4 linkage in which the number 1 carbon of the galactose is attached to the number 4 carbon of the N-acetyl galactosamine sugar molecule or the sugar molecule on the paraglobus side or your precursor substance. And one more distinguishing characteristic is that for your type 1 chains, secreted substances are glycoprotein soluble substances, while type 2 chains are glycolipids and can also be glycoproteins and glycosphingolipids. Okay, this class was actually a board exam recall question from my batch. The question asked was on the differentiation of the precursor chain types as to glycoproteins and glycolipids. And to make it more retention specific, just remember that type 1 is glycoproteins and type 2 is glycolipids as keywords respectively. And also when you will be asked the same question on which one is glycolipid, knowing that RBCs are associated with type 2 chains, you can you can just always recall that the basic anatomy of your red blood cells as lipid sandwich or lipid bilayer or lipid structure. Just like any other living cells, it is composed with a hydrophobic internal structure and a hydrophilic outer structure. But above all this, it shows that there is only a slight difference between the two precursor chains and does not significantly change the antigenic structure of your or antigenic structure and antigenic nature of your A, B, and H antigens made from different precursor substances or chains. Okay, this is a nice illustration of the beta 1,4 linkage of the type 2 precursor chain, which is primarily prevalent or predominantly seen or observed on your red cell antigen structures. I actually got this nice picture and the rest of the succeeding illustrations and from the former illustrations from our reference textbook on the modern blood banking and transfusion practices by Dennis Harmony. So as you can notice that the carbon 1 of the terminal galactose is attached or linked to the carbon number 4 of your N-acetyl galactosamine, and that it characterizes your antigenic orientation in a beta 1,4 linkage fashion or expression. Your ABH antigens develops early in the fetal life, but they actually increase very slowly in concentration during the gestational period or during the stages of pregnancy. Okay, class, I need you to listen carefully to these pointers because these are one of the most favorite questions during the board exams. Your red cell ABH antigens arises and develops as early as the 5th week of fetal life or on the 37th day of fetal life or during the second trimester of the gestational period or gestational period, which means on your second trimester of the pregnancy. Remember that all these three items are correct. However, if they were all on separate choices during your board exams and none of the choices is all of the above, chose the letter corresponding to the 37th day of fetal life because this is the most definitive, specific, and non-range period bracket. So, moving on, since these antigens are said to arise and develop as early as the fetal life, it means therefore that your ABO group antigens are already present at birth. Remember that ABO group antigens are already present at birth. And a newborn accounts for about less than 50% or 25 to 50% of the antigenic epitopes or sites found on adult RBCs. So again, class, both less than 50% and 25 to 50% are correct answers. However, if it's in a board exam case, always choose the most definitive and specific choice. So in this case, if both are in separate choices, chose the less word descriptive or less qualitative and more quantitative answer, which is your 25 to 50% numeric range value. So from our previous slides, we have explained into detail your antigen formation and development. Excuse me. We have learned that ABH antigens, and in addition to their presence on your erythrocyte membranes and in soluble form in various body fluids or our body secretions, these ABH antigens are also present in variable amounts or concentrations on your endothelial cells, your epithelial cells, platelets, and lymphocytes. And this is exactly why that in transfusion practices, an organ or in yes, transfusion practices and organ transplantations. It is a highly mandatory requirement that the recipient and the donor must be confirmed as ABO compatible before undergoing or performing the said medical procedures. Okay, here is a nice topic inclusion in a more microscopic detail explaining your ABO antigen formations into a more molecular biology concept. So your ABO gene is located on the long arm of your chromosome 9. And your chromosome 9 actually consists of 7 exons, and the last two exons, particularly your 6 and 7th, are actually responsible for your enzyme determinations or your glycosyltransferases determinations. So, class, in this concept, it says that your ABO antigens genetically arise from a single ABO gene mutation. Two of the mutations are substitution reaction, and when we say substitution, it always has something to do with the amino acids and amino acid sequences. So now the action of this mutation initiates the change of the glycosyl transferases from A enzyme into B enzyme or vice versa, while deactivating the other enzyme depending on the gene or genes inherited by the progeny from its parents. So moving on, the enzyme specific for each gene will now yield for the transfer of specific carbohydrate molecules onto the terminal ends of the paragloboside of your precursor substance, resulting to the specificity of your A and B antigens, which are termed as your immunodominant sugar molecules. Okay, class, try to correlate this with our previous discussions on the basic formation of your ABO antigens. And lastly, the deletion occurred on the 5' domain of the coding region results to an absolute inactivation of both enzymes, which results to the unchanged or unmodified H precursor molecule from your blood group O individual. So, class to try to correlate the previous discussions on the ABO antigen development and formation with this molecular genetics concept. So, from all this, we can deduce that red cell antigens A and B is caused by a substitution, and red cell antigen is caused by a frame shift mutation. The frequency of ABO blood groups. So, as per statistics, although there is a yearly or annually update as to the numerical approximation values of your ABO blood group populations, their ranking as to the frequency is never changing, meaning to say that group O is still the most common blood group in the world, followed by blood type A, B, and group AB respectively, having AB as the rarest among the blood groups. And based from our discussions on the genetic formation and development of your ABO antigens, along with their interaction of your ABO genes, has actually something to do or has a great influence in the determination of the frequency of ABO blood group populations, including the individual characteristics of your genes, enzyme efficiencies, and so on. I also no longer included the ABO blood group population distributions on different populations in the United States, which was actually included from our reference textbook, Dennis Harmoning, since it doesn't actually concern the Philippines at all. However, I'll be giving that as your reading assignment since that is quite objective and easy to apprehend. So moving on, we have your ABO antibodies. But first, I presume that everybody has already been educated about what antibodies are and antigens are, including what plasma and serum and red cells are. So when we talk about the ABO blood groups, take note that the antigen we are referring to is the one incorporated in our erythrocyte membranes, or the one embedded in our, not embedded literally, the one on the RBC surfaces, which was recently discussed. So it means that these antigens are present on the red blood cells, while your antibodies are always retrieved and are always present in your serum and plasma samples. But in blood bank class, serum and serum samples are actually the most frequently utilized, since we all know that plasma samples are obtained via anticoagulated tubes containing chelating agents that may interfere with the complement. So, moving on, the production of an individual's ABO antibodies is actually directed against the antigen or antigens that is or are absent on that individual's RBCs. Meaning to say, type of the type of ABO antibody is determined by what red cell antigen and an individual lacks. So we have already discussed your red cell antigens along with their formations and genetic makeup. Remember that group O individual, okay, starting with the table, does not code for any glycosyl transferases, which results to the inability to produce immunodominant sugars that will constitute your specific A and B antigenic structures. And this makes it an autosomal recessive trait. incapable of producing A and B antigens. And that's exactly why a group O individual lacks both A and B antigens on the RBCs. And since the production of your ABO antibodies is directed against the antigen that is lacking on the RBCs, it means therefore that the antibodies produced and are present in the serum of a blood group O are both the A and B antibodies. And the same concept goes with your groups A, B, and AB. So for your blood group A containing only A antigenic structure which lacks B antigens on the RBCs, it means that the antibody present in the serum or plasma is B antibody. While group B that only contains B antigen and therefore a antibody is present on its serum or plasma. But in case of your AB or blood group AB, it exhibits both of the A and B antigenic structures on its RBCs, meaning to say it does not lack any of the antigens. So therefore, no A and B antibodies are produced and are found in your serum or plasma for group AB individuals. Okay, class, the concept that was previously discussed leads to the explanation of these two Lancini rules that are specific only for your ABO blood group system. So take note that these two rules are actually the foundation and the most basic concept of the ABO blood group system. It also makes that the antibodies present in serum predictive and vice versa. It makes the antigens present in the red cells predictive. Now, this rule states that a person does not have antibody to his own antigen, but a person has an antibody to the antigen he lacks. And why exactly? Okay, just a bit of review on the fundamentals of your immunologic response or immune system. So an antigen-specific antibody is produced when the human body has a past exposure or that was previously sensitized to an antigen. So these built antigen-specific antibodies will now serve as a long-term line of defense against that same antigen. And if it happens that when the individual has contracted the same antigen again, as the nature of the antibodies will readily bind with that antigen, causing agglutination and triggers the immune response system or your immunologic response cells that will recruit cytotoxic cells to kill or lyse that specific antigen as a form of the defense mechanism of your body in eliminating substances recognized as foreign or non-self, which are your antigens. And the same concept applies with your ABO antigens and antibodies. Remember that for example, a person is a group A, it contains A antigen in his red cells and B antibody is in his serum or plasma, meaning to say, group A individuals has an antibody that can readily destroy B antigens. And that explains why a person does not have antibody to his own antigen because that means an auto death of the person. But however, a person with antibody to his own antigens does not actually exist much in blood bank. But one best example is when a group A person was mistakenly transfused with a group B cells. Since it is recognized as non-self or the antigen of specificity of the anti-B, it will instantly cause a rapid intravascular hemolysis of the donor cell, which will eventually lead to death or mortality of the recipient. Your ABO antibodies are predominantly IgM in nature, which is a cold reacting antibody and can readily activate your complement. And it has an optimum temperature of reactivity at room temperature or colder. So your ABO antibodies were first believed to arise without prior red cell or antigenic simulation, thus the term naturally occurring. However, given the latest scientific evidence to its production, it explained that ABO antibodies have actually occurred via stimulation of chemically similar substances to your A and B antigens that are actually present in or can be found in our nature or in our environment, such as bacteria, pollen particles, and others to name a few. So these were believed, postulated, and proven that have sensitized your body instead of your red cell antigens to produce specific ABO antibodies to antigens that your RBC lacks. Okay, additional information. Well, the actual experimentation actually conducted with chickens as experimental subjects. So when the chickens were grown in an experimental germ-free environment, they failed to develop the naturally occurring antibodies, and this experiment was actually linked with humans and was noted that human isoagglutinins or your ABO antibodies were developed the same way. So in this is exactly the reason why the ABO antibodies generally takes 3 to 6 months for newborns to develop sufficient levels of these antibodies to be readily classified in the ABO grouping, and the peak production of your ABO antibodies is remarkable within 5 and 10 years of age according to Dennis Harmening, but in some references between 2 to 4 years of age. However, when classic, note that when these two came out in your board exams, chose the former, which is from Dennis Harmening, since her book in transfusion practices is the medtech's Bible for immunohematology. So therefore, all this scientific evidence as sooner it was regarded that non-red cell simulated antibodies instead of naturally occurring became the more appropriate term to describe the production of your ABO antibodies. Okay, class, take note since it takes for about 3 to 6 months to develop ABO antibodies, which means undetectable after birth or onwards. Serum antibody typing for newborn is never recommended. Take note, the serum antibody or reverse typing for newborn is never recommended. And the same way with your cord blood samples, since most of the antibodies present are still your maternal antibodies. So, ABO antigen antibody detection. I believe that your forward and reverse typing are your first topics on your blood banking laboratory subject. However, on these following slides, we will only tackle a brief overview of your ABO antigen and antibody detection procedures, since the more complex discussion along with the procedures are designated on your laboratory subject. So in blood bank, most common requisition sent is your ABO typing, and these procedures can be classified into your forward and reverse typing. Beginning with your forward typing, okay, different terminologies are also associated with your forward typing, such as front typing, direct typing, forward grouping, cell typing, and cell grouping. But despite of the many terminologies used, they all fall as the same procedure, and that is detecting the antigen or antigens present on the RBCs or your red blood cell surfaces. So from our previous slides, remember that it was mentioned that the ABO grouping of an individual is determined by which antigen is present on his red blood cells. And that same exact statement is the main procedure concept or main procedural concept of your forward typing. It detects the presence of red cell antigens via known anti-A and anti-B sera, which are reagents used in the blood bank are mostly monoclonal antibodies that are commercially prepared solutions from animal origin. It comes with two different solutions for your antigens A and B respectively. So remember, these are antibodies, meaning they will agglutinate their antigen of specificity, meaning to say they will agglutinate when the specific antigen is present on that red blood cell. So your anti-A reagent will agglutinate the red cells containing the antigenic A determinant, while your anti-B reagent will agglutinate the red cells containing the antigenic B determinants. So for those who have tried visiting a laboratory or have had the opportunity to volunteer work in a diagnostic lab, you may have seen the anti-A and anti-B sera. Your anti-A appears in a clear blue solution due to the thymol blue dye or bromphenol blue dye used, while your anti-B appears as a clear yellow solution due to the dyes used like acliflavine or the other one, tartrazine. So next is your reverse typing, back typing, indirect typing, or serum typing. So from the previous slides, we have learned that serum or plasma samples contain, when present, your ABO antibodies. And therefore, this is the typing procedure uh, detecting the presence of antibodies in serum or plasma via known A1 cells and B cells. And these cell preparations are actually made from 45% red cell suspensions from people with known A1 and B cells. So basically, these suspensions are red cells, meaning to say they contain your specific antigenic determinants or your antigen structures which will react to your serum sample when the specific antibody is present. So, okay, class, let's review. This is an important board exam pointer, so make sure to listen, class. Your serum contains antibodies, which is basically the one we are detecting in the reverse typing with the use of your known red cell antigens. And remember that newborns develops a detectable ABO antibody titer during or at the age of 3 to 6 months after birth. And this is the main reason as to why newborns or as to why reverse grouping is never recommended. At the same time, your cord blood samples is not also an absolute sample type for newborns since it predominantly contain dilutions with the maternal antibodies. So, okay, this is the complete summary of the reactivity of each of the blood groups to different reagents and red cell suspensions utilized during your ABO forward typing and ABO reverse or serum typing respectively. Given in this table, you are your forward typing for the detection of red cell antigens and reverse typing for the detection of ABO antibodies. The sample used for forward grouping is your red blood cells since they contain antigens, while for reverse grouping uses serum samples since antibodies are present in your serum samples, may as well be used your plasma. So in forward grouping, we use antibodies as reagents, while reverse typing, we use suspensions of red cells containing known antigens. The basic principle for both ABO grouping procedure is the formation of an antigen antibody complex that causes a visible agglutination, which represents a positive reaction. So, reviewing our class Lassiner rule, a person does not have antibody to his own antigen, and a person has an antibody to the antigen he lacks. So, for a group O person, he has no antigen present on his RBCs, which means his red cells will not react to the anti-A or anti-sera A and anti-sera B under forward typing, but produces or shows the presence of A and B antibodies, which are the antibodies for the antigens he lacks. So it will demonstrate agglutination with the A1 and B cells under the reverse grouping. So remember class, that a blood type O individual does not contain any antigens on his red cells. And that's the main reason why it is the universal donor. As for a blood group A individual, it contains a antigen, so it will demonstrate agglutination with your anti-B, anti-A in the forward typing, while it will demonstrate agglutination for your B cells in the reverse grouping due to the presence of the B antibody. So group B individuals contains antigen B, so his red cells will exhibit agglutination with your anti-B under the forward grouping, while it will agglutinate A1 cells in the reverse typing due to its presence of the A antibody. To conclude the list for a blood group AB, individuals with this blood group, their red cells agglutinate with the anti-A and anti-B in the forward grouping because they contains both A and B antigens. So, uh, since they have both antigens on their RBC membranes, they will show a negative reaction for the A1 and B cells in your reverse typing, meaning it does not have any antibodies present in their serum samples, and this is exactly why a blood group AB is the universal recipient. So with this data of information, we can deduce that there is an inverse reciprocity between the forward and reverse grouping, and that they serve as counter checks for both of their results to validate the true blood type of an individual. So at this stage, I believe most of us is already acquainted with the nature of antibodies, which are highly definitive of the initiation function for the immunologic reactions to take place against any cellular or non-cellular components recognized as non-self. So the same immunologic perspective actually goes with that of your ABO antigen antibodies, particularly your ABO antibodies. So just like most of antibodies, they are highly specific against their antigen of specificity. So, okay, given that a person should not have antibodies to his own antigens, but instead has antibodies to the antigen he lacks. So it means that this antibodies has a high degree of reactivity or specificity to the mentioned antigens. So when exposed to these antigens, they will result to a rapid intravascular hemolysis, and that's the main reason why transfusion of the wrong ABO group is the leading cause of death in a hemolytic transfusion reaction. Having specifically TRALI or your transfusion-related acute lung injury is the most frequent cause of death. And this is exactly the main reason why before your transfusion or blood transfusion, it is mandatory to make sure that the donor and recipient are ABO compatible. So as you can notice, I have included here the cellular mechanism of how the TRALI affects your alveolus or your lung cells. However, it has a complex cascade of cellular interactions that may require for us to have a separate discussion. And well, of course, it will be tackled in a separate designated week in our immunohematology lecture. Moving on, so aside from your monoclonal antibody reagents for the forward grouping, blood banks also utilizes a different source of antibody preparations for the detection of your ABO antigens. So these are actually plant origins, which is particularly your plant lectins. So lectins, defining them, are biochemically identified proteins that are present from the plant seeds, and their action is similar to your ABO antibodies, where it binds specifically to the carbohydrate determinants or your immunodominant sugars present in your red cells. And the positive result is the presence of an antigen antibody binding or agglutination when the specific red cell antigen is detected. So, okay, class, take note that this is a board exam pointer. Okay, in blood bank, we use three different lectins of plant origin. The anti-A1 lectin that agglutinates A1 or A1B antigen but do not agglutinate A2 or A2B antigen. As you can notice, so there are, I'm mentioning more often numerics of 1 and 2. These two are actually the common subgroups of A, which I'll be discussing in a more elaborate manner and in a separate topic slide. So we have also your anti-B lectin that agglutinates B antigen and anti-H lectin that agglutinates your H antigen. So your anti-A1 is from the plant Dilocos biflorus. Your anti-B is from Griffonia simplicifolia, which is formerly known as Bandereia simplicifolia, while your anti-H is from Ulex europaeus. So remember class, your formation and development of the ABH genes or your O gene, which I keep on reiterating multiple times, do not code for enzyme production resulting to unchanged H antigen in the red blood cells of the for red blood cells of the blood group O individuals, meaning to say they contain the highest concentration of H antigens in their red blood cells. And that's exactly why, as you can notice here, I've included here in this particular slide the reactivity and frequency of your H antigens in different blood groups. So your H, your O antigen, your O blood group O is the most abundant of H antigen. So meaning to say it will react the strongest with anti-H lectin, and then followed by your A2B, 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 A1, and A1B with the weakest reaction to the anti-H lectin. So I will be explaining this further on the as we proceed with the discussion. So next, as I was mentioning a while ago, you have the A1 and A2 common subgroups, but your blood group A actually consists of the two subgroups, and namely A1 and A2, and six more other common subgroups. So meaning to say there are plenty subgroups under your blood group A. Beginning the discussion by introducing you first with the molecular view and concept of the interaction and conversion efficiency of your enzymes. So basically, all of these illustrations are A antigen examples, building the specificity for blood group A. So as you can notice that there are four different structures of the H antigens, which are H1, H2, H3, and H4 respectively. But despite of the varying differences in chain types, you can notice that all of them exhibits the same terminal sugar molecules, which is your N-acetylgalactosamine, which is the immunodominant sugar for your A antigen. So on your lower left table shows the quantitative and qualitative difference of your A1 and A2. And one of their qualitative distinction is the subtle difference on the transferase enzyme, specifically your N-galactosamine transferase. So both of A1 enzyme and A2 enzymes can efficiently convert H1 and H2 antigens into AA and AB determinants respectively. However, your A2 was recorded to A2 enzyme was recorded or remarkably less efficient as compared to your A1 enzyme. Meaning to say, A2 enzyme can convert H1 and H2 antigens but in lesser amount as compared to your A1 enzyme. And that is evident actually on the quantitative difference, that there is a decreased amount of the enzyme that may have resulted to a less efficiency in the conversion than your A1 enzyme or A2 as opposed to your A1 enzyme. So while your more complex H3 and H4 antigens are converted efficiently by A1 enzyme into AC and AD determinants, while in case of A2 enzyme, conversion is remarkably poor on H3 and failure of conversion of for H4. And this is exactly why A2 subgroup contains higher concentration of unconverted H antigen sites than your A1 subgroup. However, it appears to be a less branch structure as opposed to your A1. On your lower right box shows the summary of the structural characteristics of your A1 and A2 subgroups. So overall, basing from the conversion efficiency of A1 enzyme, it actually resulted to A1 subgroup containing all A antigen determinants and that left zero unconverted H3 and H4 antigens, while A2 subgroup are predominantly A and AB determinants only, with a remarkably poor amounts of AC determinants and a complete absence of your AD determinant, which is actually which actually accounts for the very few individuals that may produce an unexpected anti-A1 antibody in that individual's serum sample. So moving on, we have here the summary and a simplified version of the differences and main characterizations of your A1 and A2 phenotypes or your common subgroups. These two common phenotypes were actually first discovered or described by von Dangern in 1911. So included in this table are your reagents, which includes your anti-A, anti-B, and anti-A1 lectin, also your special polyclonal antibody, anti-AB. This is polyclonal since it has multiple antigenic specificities, meaning to say it will agglutinate in the presence of an A antigen, and it will agglutinate in the presence of B antigen, or it will present agglutination or a positive reaction if both antigens are present. So we also have here your common and unexpected antibodies found in the serum sample of an A1 and A2 phenotype, including also other distinguishing parameters like the soluble substances present and the number of antigenic sites present on the red cells of your A1 and A2 subgroups. Both okay, now class, let's start from the table. Both phenotypes exhibits a strong agglutination reaction on the anti-A, and since both are still blood group A, it means therefore that they lack the B antigen, resulting to a negative agglutination on your anti-B reagent, while the expected or common antibody present in serum is predictably your anti-AB. So that's one more practical application of your Lens-Linear rule. Okay, class, take note that your result in A1 lectin, anti-A1 lectin is actually the most important parameter in distinguishing between A1 and an A2. So it is already highlighted on the table. It is because a true A1 red cells is agglutinated with your anti-A1 or your Dilocus biflorus lectin, remember, while leaving your A2 red cells unagglutinated. And this is actually the result from the previously discussed the efficiency of the A1 enzyme over the A2 enzyme in successfully converting all the H antigen glycolipid chains into an A antigen determinants A, B, C, and D that actually codes for A1 antigen specificity, which was freshly discussed from our previous slide. So, in other descriptive terms, it has created a high concentration of A1 antigenic specificity, which also makes it more reactive with the anti-A1 lectin. Excuse me. And the agglutination on both anti-A reagent and anti-A1 lectin determines your A1 subgroup, while agglutination only on your anti-A reagent determines your A2 subgroup. So, class, as I have mentioned from the previous discussion, that A2 individuals produces trace concentration of the antigen C determinant that may still initiate the production of very small amounts or concentration of an unexpected anti-A1 in the serum sample. So, which now accounts for the very rare case of 1% to 8% probability case occurrence from A2 individuals. So, okay, given the data, approximately 80% of group A or AB individuals are A1 or A1B, while the remaining 20% accounts for those group A2 or A2B. So, okay, class, one more important thing to remember. that I have mentioned in our earlier slides that in a group AB individuals, B is more competitive than your A gene at age determinant size. And since subgroup 2 is weaker subgroup than A1, it means therefore that in a group A to B individual potentially results to a more jeopardized A to conversion efficiency since B gene will dominate the H antigen sites. So it means therefore that a complete absence of both A antigen C and D determinants in a group A to B. And by following the Landsteiner rule, and since A antigen, A, B, C, and D became determinants of A1, and that A2B lacks these C and D determinants, meaning to say A2B will undergo a consequent production of antibody for the antigen that he lacks and therefore produces traces amounts of anti-A1. And that now accounts for the 22% to 35% of A2B individuals that produces anti-A1. So I hope that everyone is diligently following the discussion. So, given the previous discussions and additional information tackled about the common A subgroups, here is a quick recap of the abundance of H antigens of each of your blood groups and their strength of reactivity to your anti-A1 lectin. So again, your blood group O among all ABO blood groups contain the most abundant H antigens because it does not code for the glycosyl transferases, giving the inability to convert the H antigen, meaning to say it contains a very high concentrations of your unchanged H antigens. Your blood group A2, as previously explained, exhibits poor enzyme efficiency compared to A1 enzyme and poorly converts your H3 antigen and a failure of conversion on your H4 antigen, resulting now to a high amount of this unconverted H3 and H4 antigenic sites. Okay, followed by your blood group B, which is known to have a variability of its conversion and of the H antigens. However, it is placed third among the blood groups for anti-H reactivity. So now, this was actually recently discussed. Your A2B exhibits complete absence of the AC and AD determinants due to the unconverted H3 and H4 antigens. So remember that your A2 has a characteristic weak enzyme activity that is only capable of converting H1 and H2 antigens with poor conversion of H3 antigen. And while your B has a variable reactivity, and that when coupled with your A antigen, I mean A gene, becomes more competitive at H determinants and will tend to dominate over your A2 enzymes. But given this circumstance, however, the conversion of your H3 and H4 antigens actually resulted to a detectable H antigen concentration that are mostly H3 and H4 determinants. Moving on with your blood group A1, since this is actually the subgroup of blood group A that elicits high concentration of glycosyl transferases, specifically your N-acetylgalactosamine transferase, that results to the converting almost all H antigenic sites available on the red cells. So at this point, it is very obvious that A1B has the least amount or concentration of your H antigens, and that will react the weakest on your anti-H lectin, since both A1 and B are highly competitive for the available H antigen sites that will convert almost all H determinants. And that's exactly why it exhibits the least amount of H antigenic sites among your ABO blood groups. So with your weak A subgroups, it only accounts for 1% case or less of case occurrences that are encountered in your blood banks and are actually not given much of importance in the clinical field. But given the rarity of the case, however, it poses a great deal of academic interest and is always a favorite topic of your board examiners. That's why it is always emphasized in teaching institutions and most particularly during your exams on your board exam review centers. So I am showing you now is the main summary of the 6 week subgroups of blood group A. So at first glance, this table appears to be quite overwhelming, and I hope that everybody is diligently following with the discussions, since understanding from our previous discussions is the key to understanding these concepts. So take note, class, and remember that in a typical group A, it exhibits reactions on your anti-A, anti-sera, while reactivity on your anti-B in the serum samples in reverse typing. So it is imperative to observe on these two parameters and try to correlate with that of a typical group A. So okay, moving on. This table shows the reactivity of each of the weak A subgroups with different reagents and rest of suspensions, along with additional parameters like your soluble substances in saliva, your presence of AH transferase in serum, and number of antigenic sites formed on each of the weak subgroups. Okay, class. MF represents mixed field reaction, meaning small agglutinase within predominantly unagglutinated red cells. Zero represents negative reaction, meaning no agglutination reaction occurred and shows the absence of that parameter with the given weak subgroup. So on your and your 2 plus to 4 plus represents the degree or strength of the agglutination reaction and shows the presence of the given parameter with that specific weak subgroup. So starting with A3 red cells. The hallmark characteristic of A3 is the mixed field reaction on your anti-A and anti-AB anti-sera. The estimated antigen size per individual RBC is approximately 35,000, with a non-definitive presence of A transferases in their serum samples. Your AX red cells generally exhibits no agglutination with anti-sera but shows reactivity with your anti-AB. Take note that in AX, anti-A1 is always almost present. So for your Aend demonstrates agglutination, however, only exhibits a weak mixed field reaction on both anti-A and anti-AB sera, where only 10%, okay, remember class, only 10% or less of the RBCs were agglutinated. It contains about 5,000 antigenic sites per individual RBC. And take note, class, that your antigen sites has something to do with the enzyme efficiency or the conversion of the H precursor substance. So for AMRBCs, they are mostly unagglutinated with your anti-A and anti-AB, but in less probable cases may demonstrate agglutination but very weak reactions only. And it does not also agglutinate your anti-A1 lectin. So for AY RBCs, they demonstrate negative reactions for anti-A and anti-AB. And your AY red cells are predominantly the result of a recessive mode of inheritance. Particularly example would be in a sibling or in siblings. And since it's recessive, it means therefore that it does not represent any expression of an alternate allele at your ABO locus, but a germline mutation of a family member. To complete the list is your AL red cells. Just like AM and AY, they does not generally agglutinate or react with your anti-A and anti-AB sera. So it demonstrates the least amount of antigenic sites produced. Okay, class, just a smart study tip when dealing with this kind of data of information, most particularly when doing your board exam review or any other exams. Taking this as an example, always observe the parameters where a certain subgroup is unique about, among others, or the details that makes the hallmark that characterizes them among from the other subgroups, or via grouping them with their similar identifiers or serologic pattern of the activity. So let's take a look at this present slide. So applying your Landsteiner rule of the antigen and antibody presence, since this is a subgroup, right? Meaning to say it must be negative for anti-B anti-sera, while positive on B antibody in your serum sample. Moreover, it must demonstrate agglutination on anti-A anti-sera and anti-AB anti-sera since it contains the A antigen. However, this isn't the case for AM, AY, and AL, and that's exactly why they are subject for elution and adsorption procedures. So for your adsorption elution, a bit of review since this will be discussed in your BB lab. Okay, this procedure is utilized primarily for those that fail to exhibit agglutination on their expected anti-sera of agglutination. So meaning to say, since it is blood group A, they are expected to have agglutination on anti-A in the forward grouping. However, in the case of AM, AY, and AL, they demonstrate absence of agglutination, and that they may be believed, they may be believed to have absorbed the antigen on the red cell surface, and their presence can be confirmed only using, or it can be confirmed only via your adsorption elution technique. So basically, class, the action of this technique will free the antigen from the red cell and exposing the antigen. Therefore, take note that serum agglutination results of AM, AY, and AL can only be achieved via your adsorption elution procedures. So moving on, remember that your AY is characterized by being a result from a germline mutation of a family member. So, okay, take note that mixed field reaction of both anti-A and anti-AB anti-sera is the hallmark of an A3 red blood cell. On the other hand, Aend also demonstrate mixed field but in a very weak reaction only, since anti-A and anti-AB only agglutinates about 10% or less of the Aend RBCs, and was also associated with two known variants, namely your A-Bantu and A-Fin, which is not given much of importance, but it is good to know. So for your soluble substances in saliva, only A3, AM, and AL produces both of the H and A soluble substances, while only A and AL red cells show a complete absence of A transferase in their serum samples. And lastly, your AM and AY red cells demonstrate a complete absence of the anti-A1 in serum sample, and that is for your weak A subgroups. So based from the previous slides, these listed techniques are utilized for the investigation and differentiation of your weak A phenotypes or subgroups. For the forward grouping, we detect for the presence of your H and A antigens present on the red cells of your weak A subgroups. Reverse grouping for ABO antibody detection and for the presence of anti-A1. Saliva studies for your detection of the A and H soluble substances for secretors, and the adsorption elution procedures specifically for AY, AM, and AL. And investigation and differentiation can also be achieved via molecular assays and enzyme studies, or with the enzymes responsible for your immunodominant sugars. Similar to your weak A subgroups, your weak B subgroups can also be differentiated and investigated via these following techniques or procedures. Okay, testing the strength and the type of agglutination with anti-B, anti-AB, and anti-H in the forward grouping, the presence of ABO isoagglutinin or your ABO antibodies with the use of your reverse typing, and the soluble substances produced in the saliva, which also determines if they are secretors or non-secretors, and lastly, your molecular studies. So just like your weak A subgroups, weak B subgroups demonstrates the same pattern of reactivity, and that's why, as you can notice class on this table, the phenotypes are almost the same with that of your weak A phenotypes, only that the letter A is replaced by capital B. However, in this case, there's no recorded similar pattern of reactivity recorded for your AY and AN, and that's exactly why we only have here B3, BX, BM, and BL. Okay, starting with B3. Okay, B3 red cell is the most frequent among your weak B phenotypes, and the same hallmark with your A3. It also is characterized by mixed field agglutination, but only for anti-B, of course, since it is weak B subgroup, and anti-AB, anti-sera. So it does not demonstrate an expected antibody in the serum sample. So while your BX exhibits only a very weak agglutination reaction on both of your anti-B and anti-AB in the forward grouping. So take note that, as you can see in this table, it may produce a weak concentration of anti-B. So recalling our Lanciner rule, excuse me, it clearly is an exemption, and why exactly? So, because it only exhibit very small concentration, your body fails to recognize the weak antibody as non-self. And given the extreme rarity of the case, it may otherwise not given much of attention, and so there's no much of records actually to expound into detail. However, take note that BX shows a complete absence of the B enzyme. So next is your BM red cells. As you can see, class, they are generally agglutinated by your anti-B and anti-AB in your forward grouping. BM subgroup contains your BM transferase in serum, however, in a very low concentration. So, okay, class, just an additional information. Your BM is actually a result of a decreased transferase activity in your hematopoietic tissues. It was the proven cause by a Japanese scientist experimented on a normal B plasma mixed with your BM weak subgroup with the addition of your uracil diphosphate galactose. And this experiment actually enhanced the enzyme activity by creating a synthetic replacement of the immunodominant sugar and transformed your BM subgroup into a normal B phenotype. And okay, to conclude your BM subgroup, take note, class, it has case frequency that was recorded in Japan. So lastly is your BEL, or your Bel. It almost has the same reactivity with your BM, having an agglutinated by anti-A and anti-AB, anti-sera. So unexpected weak antibodies are present in this room, which is anti-AB, and the only difference was demonstrated on the soluble substance, with BL having only H substance present and the complete absence of the B transferase. So, okay, class, take note that the hallmark for BEL is its unique mutation location. Remember that coding regions for the ABO transferases is at the 6th and 7th exons of your chromosome 9. And most mutations in the ABO occur in both 6 and 7 exons. However, in the case of your BL subgroup, so the mutation has occurred only at your 7th exon of the ABO loci. Acquired pseudoantigens. This occurs when bacterial enzymes or polysaccharides try to modify the immunodominant sugar determinant sites on your red blood cells via absorption of bacterial polysaccharides, which results to the creation of biochemically similar copies that may resemble or resembles your immunodominant sugar carbohydrates that builds your red cell antigen specificity. So, class, these are board exam emphasized topics, so take note and listen carefully. Pseudoantigens, from the term itself, pseudo meaning false or a lie. These are particular substances of bacterial origin that biochemically resembles your red cell antigens, specifically your A and B antigens as to their carbohydrate, oligonucleotide chain structures, or the immunodominant sugars of A and B antigens. And what we mean by that is it also therefore mimics their reactivity to your anti-B serum, most specifically during your forward grouping, which means that they will result to false positive reactions or cross-reactivity on your forward typing. So two clinically significant pseudoantigens are very famous during board exams and board exam reviews are your acquired A and acquired B antigens. Actually, class, acquired pseudoantigens appears to be of iatrogenic cause of your gastrointestinal origin, meaning when your normal physiology is altered due to the certain conditions like underlying carcinoma of the colon, gastrointestinal infections, GIT or gastrointestinal obstruction, also septicemia or any other intestinal disease that results to the dramatically slowed gastrointestinal motility, which now facilitates the portal of entry of your GIT normal microbiota to your bloodstreams via intestinal capillaries or your lacteals. And we'll be discussing this in just a moment. So for acquired A antigen, it is reported in persons of type O or B with predominantly biologic agent Proteus mirabilis, while acquired B antigen is associated with blood groups A individuals having Proteus vulgaris and Escherichia coli serotype 086. So we have here is a nice illustration of your magnified view of an intestinal villus. It is also a review from your anatomy and physiology. I recall I got this picture from your Anaphysio by Gerard Tortura just for a rough representation of the cellular networks that are actually involved in the absorption of your bacterial polysaccharides and enzymes. So as I was saying a while ago, the presence of any sort of gastrointestinal disease that results to a dramatically slowed gastrointestinal motility is actually the main problem. So we all know that the final digestion and fluid reabsorption takes place on your in your colon or large intestines, and that we actually have billions of normal microbiota in our gastrointestinal tract. Now, when a disease or infection is present, like in carcinoma, intestinal obstruction, or heavy infections, it will alter the physiologic movement or motility of your intestines, and it will result for the undigested fecal matters and waste substances to stay longer in your colon than in your normal physiology. And since colon is the final site for your fluid reabsorption, which is actually regulated via osmosis, which means that there's no negative feedback loop to actually control or stop the water reabsorption function of your colon. So now, the longer these fecal matters stay in your colon, the more and more and more fluid or water is reabsorbed back to the capillary networks and lacteals via albumin transporters. And what comes with water are your electrolytes. And since there is a dramatically slowed gastrointestinal movement and a prolonged stay of these fecal matters, there goes your accidental absorption of your bacterial polysaccharides that will now eventually pass on to your blood system and causes desensitization or modification of your immunodominant sugars on red cells, which actually involves another complex cascades of cellular recognitions and etc. But cutting it short, class, the bacterial material absorbed actually deacetylates or removes the acetyl group from your N-acetyl galactosamine, converting it into D-galactosamine, which is biochemically similar to your D-galactose. And this modification will now be carried by your erythrocyte membranes. However, it will not be targeted by your cytotoxic cells via your B antibodies, since it will not be recognized as non-self. And in blood banks, it normally causes a weak or mixed field agglutination reaction with your anti-B, anti-Sera in the forward grouping. And that is exactly why in an acquired B phenomenon, if the person is group A, and due to the presence of the bacterial modification that mimics the B antigen immunodominant sugar, this person's red cells will react to anti-B sera in the forward grouping, resulting now to having a blood type AB. And the same concept also goes with your acquired A phenomenon. If a person is blood group B, his red cells will type as AB in the forward grouping, and group O into group A respectively. And above all, class take note that for these two pseudoantigens, they only cause discrepancy in vitro, meaning to say, since these acquired A and B antigens does not actually stimulate for any or intracellular or vascular immunologic response, meaning to say they only exhibit false agglutination reaction, most particularly in your forward grouping, while the true blood group or blood type can be revealed via your serum antibody typing. Next is your Bombay phenotype, the H null or H deficient phenotype. It was first reported in Mumbai, India in the year 1952 by a scientist named Bende. So let's recall that from our previous discussions on the inheritance of your ABO genes and antigen formations, we have learned that the inheritance of a dominant H gene is crucial in the formation of your antigens since it codes for the glycosyl transferase that facilitates the placement of your L-fucose sugar to the terminal galactose of the paragloboside to form your H antigen, while your H substance will serve as the precursor for your A and B enzyme to form A and B antigens. Okay, however, class in this phenotype, it is inherited as an autosomal recessive trait that represents the inheritance of a double dose of small h gene, resulting to a very rare genotype HH. Take note that this occurs as identical phenotypes in children, but not in parents, meaning to say this occurs in a progeny of normal parents, only that this progeny has inherited a pair of recessive H alleles from the parents. So it is explained why it is called as an autosomal recessive. And just for an additional information from a cross-reference, this phenotype is expected or most likely to arise from a disputed paternity or from consanguineous marriages or marriages between closely related people, such as marrying as close as your third degree cousin. So, moreover, class, take note that an individual in this phenotype may still inherit the functional A or B genes from parents, and at the same time, A and B enzymes are still present in their serum. But well, of course, depending on which gene is inherited. However, due to the recessive inheritance of the H gene, these enzyme functions are totally impaired, meaning non-functional but present. Okay, based from this, if a Bombay phenotype has an inherited A gene, B gene or AB gene, they are written as superscripts of OH, like what you are seeing here in this particular slide, OHA, OHB, and so on. Okay, moving on. Okay, given that this is an inherited double dose of amorphic H alleles, your FUT1 and FUT2 genes that codes for the same glycosyl transferase are silenced, which will absolutely result to the absence of both ABH antigens in your red cells and ABH soluble substances in your saliva or body secretions. So, the RBCs of Bombay phenotypes does not agglutinate anti-H lectin in the forward grouping because it literally, obviously, it does not have any H antigen form. And going back with your Landsteiner rule class, since Bombay phenotypes does not produce any antigens on its red cells, then therefore it will produce antibodies directed against the missing antigens, particularly your anti-B, anti-A, and anti-H antibodies. So take note that this is one of the most important pointer in this phenotype. So since it does not form any antigens, Bombay phenotypes are typed as blood group O in your forward grouping. And your Bombay anti-H antibodies are highly IgM and highly potent antibodies that has a wide range of thermal reactivity and can instantly lyse red cells when exposed to red cells with the H antigen. Most especially, for example, when group O cells, which having the most abundant of the H antigen, are accidentally transfused in a Bombay phenotype recipient with the with a highly potent H antibodies, it will immediately lyse the donor cells, leading to rapid intravascular hemolysis and death. And that's exactly why RBCs of Bombay phenotypes are compatible only with the serum from another Bombay phenotype individuals or blood from the same phenotype. This is a perfect tabular comparison between a group O and an HNAL or your Bombay phenotype. Remember that a Bombay phenotype is mistakenly phenotyped as group O in the forward grouping, since just like your blood group O, H null phenotype also do not present an antigen on their RBC membranes, and they can only be differentiated by the use of your anti-H lectin in the forward grouping and O cells in the reverse or serum typing. Your anti-H here is an antibody specific for the H antigen, while O cell is a red cell suspension with a known group O's with RBCs or red cells, meaning to say it contains the H antigen of the O red cells and will agglutinate when exposed to a serum containing the anti-H. Take note that agglutination is the clumping or aggregation of your red blood cells due to the antigen-antibody complex or antigen-antibody interaction that shows a positive result when present. So, okay, remember also that in a forward grouping, we are detecting for the presence of antigens, while in the reverse typing, we are detecting the presence of antibodies. So, from our previous discussion, we have learned that your blood group O individual has the highest concentration of the H antigen present on its red cell membranes, and will therefore show a strong agglutination with your anti-H in the forward grouping. So, in the other hand, an individual of Bombay phenotype, the H gene is inherited as an autosomal recessive and does not have the ability to produce any antigens, which exactly means that he lacks expression of its antigen. Then, therefore, a Bombay phenotype does not demonstrate agglutination reaction in your forward grouping. So, for one more time, recalling our Landsteiner rule, a person does not have antibody to his own antigen. So it means, therefore, in the reverse typing, group O red blood cells will not agglutinate your reagent O cells because blood group O has the H antigen presented on its red blood cells, while for an H null red blood cell will demonstrate agglutination in the reverse typing because, going back with the Landsteiner rule, a person does not have antibody to his own antigen but has antibody to the antigen he lacks. It simply means that since Bombay phenotype does not have the H antigen on its RBCs, when therefore it produces your anti-H, and that's why RBCs of a Bombay person or Bombay phenotype will agglutinate your reagent O cells in the serum or reverse typing. Okay, aside from your Bombay phenotype, which is an H gene deficient phenotype, we also have here another H deficient phenotype known as your para-Bombay phenotype. So, just like your Bombay phenotype, RBCs are completely devoid of the H antigen, or in extremely rare cases, may also demonstrate but a very small amount of the H antigen. So, okay, to make it short, the genetic basis for the para-Bombays is actually a mutated FUT1 gene with or without an active FUT gene, or a silenced FUT1 gene with an active FUT2 gene. So remember that FUT1 is your H gene, while FUT2 is your secretor or secretor genes, and that both are encoders for the same exact similar enzyme, which is your L-fucosyl transferase. So the former mentioned is the one responsible for the formation of the ABH antigens on your red blood cells, while the latter encodes for the formation of your ABH antigens on your body secretions or soluble substances. So going back on the genetic basis of your Parabombay, resulted from a mutated FUT1 gene but contains an active FUT2 gene. And that's exactly why for Parabombays, unlike the classic Bombay phenotype, has a normal expression of the ABH antigens in secretions and body fluids. Okay, blood group mythology. Like it's actually one of the funniest medical book content I have ever read. You can find this in your reference textbook in Dennis Harmony. So they say the blood group A people are hangovers, which means they may probably have a low tolerance to alcohol or maybe just alcohol addicts. I just don't know. Blood group B people are associated with criminality, which is actually the worst possible judgment and unjustified characterization that a person might get from others just basing from being a blood type B. And lastly, blood group O people are associated with good teeth. But above all class, from the term itself, mythology, which means that these concepts are very far from the reality of a scientific basis because we build our own character and the establishment of one's or an individual's personality is independent of what blood type you have. And if you have blood group, for example, if you are a blood group A or a blood type A person and you practice oral hygiene efficiently, then you'll have a nice teeth. And if you're blood type B, but you are emulsified with the prudence of living, then it's unlikely for you to become a criminal. So these are the characters associated with the blood groups that were mentioned in our reference textbook. However, since they do not pose really any clinical significance and lacks a scientific basis, they were regarded just as blood group mythology. So for our last topic on this week's discussion is your ABO discrepancy. All of the non-matching results or unexpected agglutination reactions that arises and are observed on the forward typing and reverse typing that actually confuses us medical technologists in reporting the true blood type of patients are your ABO discrepancies. Okay, class, at this point, we already know about the Lensterian rule. And given that the forward and reverse typing can be counterchecks with each other's results, it is easy to determine the presence of an ABO discrepancy. So I will give you an example case. So, for example, when a person's RBCs did not show any sign of agglutination on anti-A and anti-B in the forward grouping. So based from that results, we can already deduce that the person is blood type O. However, seeing his serum typing that also did not show any agglutination on A and B antibodies is actually an example of an ABO discrepancy, since given that the results on your forward typing are not parallel or inconsistent with the results obtained from the reverse grouping. So we will be elaborating this on the succeeding slides. So first, these are the common causes of technical errors that may lead to an ABO discrepancy. Number one, inadequate identification of blood specimens, test tubes, or glass slides. So in this case, it may be a result of an illegible handwriting on the specimen label, the test tubes, and slides when performing the procedures. So number two, cell suspension either too heavy or too light. Okay, this is a probable result due to an incorrect procedural performance of the instructions given in making red cell suspensions, or maybe at the same time caused by your prozone and postzone phenomenon. So clerical errors, which may also include your improper identification of patient sample for testing, mentioned seconds ago, and the improper recording of results. For example, when there is an observed agglutination, but however you have mistakenly written a negative result instead of a positive agglutination result, then it will lead to a clerical error. So next, a mix-up of samples. This actually happens. Okay, class, just for an example case scenario, but may not be limited to. When the medical technologist is from a straight 72 hours duty, and this means that maybe she's probably tired or maybe exhausted, and that may result to the inefficiency of the medtech, particularly with the attention to detail, which can particularly result to a mix-up of samples and may also result to a misobservation of hemolysis. Or when the medical technologist is inattentive or somehow preoccupied, this may lead to a misobservation of hemolysis, failure to add reagents, and any other possible technical errors that may result to an ABO discrepancy. So failure to follow manufacturer's instructions, also similar to the one mentioned a while ago on the cell suspensions. Take note that an ABO discrepancy can also be a result of a poor laboratory equipment quality control or calibration maintenance, which includes your uncalibrated centrifuge and contaminated reagents, two factors that greatly causes unsatisfactory results. So lastly, the sample warming during centrifugation. Okay, this may have been noticed by some of you who have been or who have experienced volunteering or working in a laboratory or in laboratories. You can notice that a freshly removed glass tube from the centrifuge will feel hot, and that can be explained by the physics involved in centrifugation. And in this case, may have the probability to enhance the reactivity or unexpected warm reacting antibodies present in the sample. And above all, the first step to resolve the presence of an ABO discrepancy, most particularly when or if an error in specimen collection and identification is suspected, and also in anticipation that it may be a result of any possible technical errors, is to request for a new sample and repeat the entire procedure. And it is actually the protocol that every blood bank medtechs must follow. First thing is to repeat the entire blood grouping procedure. And if the discrepancy is resolved or the testing yielded a parallel result of both forward and reverse, the blood group or blood type of the patient or the person can be expedited or released. So, as I was saying a while ago, the first step to resolve a discrepancy or an ABO discrepancy, most particularly when if it's an error in the specimen collection and the sample identification is suspected, and also in, of course, anticipation of that it may be a result of any possible technical errors, is to request a new sample and repeat the entire testing procedure. And it is actually the as I was saying a while ago, the protocol that every blood bank medtechs must follow. Repeat collection and then repeat procedure. And if the discrepancy is resolved or the testing yielded a correlative result of both of your forward and reverse typing or grouping, the blood group of the patient or the blood type of the patient can be released or can be reported. So, however, if the same unmatching results or the presence of the unexpected reactions still occurs after the repeat testing, the medical technologist will have to search and investigate with the patient's information. Like number one is age. Okay, that should be the first one to ask or to investigate the age, followed by the diagnosis, medications, transfusions, and history of pregnancy, because any of this mentioned may have affected the ABO grouping. So, and then followed by the determining whether the discrepancy was in the reverse or forward typing via observing the weakest reactivity. So, that is a very particular class that when you can determine the discrepancy by looking to it or observing the pattern of reactivity, which is the weakest one, is the probable ABO discrepancy. Because remember that in ABO reverse and forward typing usually or almost often yields a strong grading of agglutination or a strong strength of agglutination, which is a range from 3 plus to 4 plus. So, the medical technologies may now resolve the ABO discrepancy in a different basis given different classifications, which will lead us to our next slide. So, ABO discrepancies are actually categorized into four main groups, and each discrepancy classification are actually resolved in a different approach. So, a review: ABO discrepancies occurs during forward typing, on the reverse typing, or can occur on both of the grouping procedures. Meaning to say, a discrepancy may arise if there's a problem on the patient's red cells or antibodies. In group 1 discrepancy, there is the presence of a weakly reacting or missing antibodies. In group 1 discrepancy, we are dealing with antibodies, so it means therefore that it occurs during your serum typing or reverse typing. So for group 2 discrepancy, when there is a presence of weakly reacting or missing antigens. So as opposed to your group 1, the main focus of group 2 discrepancy is the presence of weakly or reacting or missing antigens. So meaning to say, this discrepancy or the group 2 discrepancy classification occurs during your forward grouping or forward typing, while your group 3 results when there is a protein or plasma abnormalities. So remember that aside from your serum samples, plasma also contains antibodies, which means that this group also occurs during your antibody detection. So lastly, your group 4 discrepancies, which results to unexpected reactions, particularly on the serum grouping, due to the presence of cold autoantibodies and non-expected alloantibodies. Your group 1 discrepancy results from a weak or missing antibodies, so they occur on the reverse or serum typing. Given that patient information is already determined by the medical technologists, these are the main reasons and the iatrogenic causes that results to a weak or missing antibodies. Remember that in your newborns, the ABO antibody production is not detectable until 3 to 6 months of age, meaning to say they produce a sufficient detectable amounts of ABO antibody when reach 3 to 6 months of age. And this is exactly the main reason why, as I keep on reiterating multiple times, that a newborn is never recommended for a reverse grouping procedure, or that a reverse typing or reverse grouping procedure is never an option to blood type a newborn baby. So for elderly patients, so we have learned that the antibody production peaks between 5 to 10 years of age and gradually declines through age. So, for elderly patients, they already have a potentially lowered or depressed production of antibodies, affecting now the production and expression of the ABO antibodies. Antibodies will, which will demonstrate a weak reaction to a negative reaction in the reverse grouping. And actually, it same goes with their cellular and humoral immune responses or immunologic responses. So for old age individuals, it means to say that um, their immune system is declining, and that's also why, as you may have noticed during the pandemic surge class, that they are the most vulnerable. So for patients with leukemia or lymphoma, this actually has a better explanation involving its molecular biology pertaining to the mechanisms of the CD95 and ligand interactions, especially in chronic lymphocytic leukemia. However, class, to cut it short, the main characterization is that it causes hypogammaglobulinemia. So from the term itself, hypo meaning low or below normal, and gamma globulin pertains to your immunoglobulins or your antibodies or ABO antibodies. And your ABO antibodies are IgM predominantly, and IMIA from the term hypogammaglobulinemia. The IMIA is a medical extension term to denote the presence of. So constructively in leukemia, there is the presence of low production of gamma globulins or antibodies, which means, or which in the reverse typing will result to a weak or missing antibody. So the same concept actually goes with patients receiving immunosuppressive drugs like cyclosporines or what else, corticosteroids. For example, in conditions like multiple sclerosis, systemic lupus erythematosus, and Crohn's disease, and so on. And these particular conditions, their immunologic response actually are actually they actually fail to recognize their own cells as self, and that's why their immune cells attacks and kills their own body cells, and that's why they are given drugs to suppress the activity of their immune system, particularly the antibody production, which is now the side effect resulting to the hypogammaglobulinemia that also depresses the production of your ABO antibodies. Okay, you have here a same case, but this time the total absence of the antibody production, with the patients having congenital acquired a gamma globulinemia, or what we call as Broughton's disease, which is a sex-linked disease inheritance that actually occurs only in boys. That's an additional information from your cytogenetics. Gamma-globulinemia, from the term itself, incapability of producing antibodies, which results to a complete absence of agglutination in the reverse grouping. So when the patient is under recovery from a post-bone or stem cell transplantation, they will likely to demonstrate a weak or negative agglutination reaction in the reverse grouping, since their body will keep adapting with the transplanted bone marrow and stem cells. And actually, or eventually after the recovery, they will start producing different RBC populations from that of the transplanted bone marrow, with with patients whose existing ABO antibodies have been diluted by plasma transfusion, may also result to a very weak agglutination reaction in the reverse grouping. So, lastly, is the presence of ABO subgroups that has been discussed previously. Since the facts that they were characterized as subgroups, it only denotes that they would likely or predictively result to a weak or missing ABO antibodies in the serum typing. So here is a nice example of a group 1 case discrepancy. Okay, forward typing results always comes first and followed by the reverse typing result. So basically, the forward typing result is the initial basis for the blood group of the patient. And basing from this table, the probable blood type is blood type O. And remember that for blood group O individuals, they do not contain red cell antigens. Therefore, they will produce antibodies for the antigen they lack. So it can be expected that there should be a strong agglutination in the reverse typing. However, So, class, as you can see in this table, given in this case, the reverse grouping shows an absence of agglutination on the reverse typing or reverse grouping. So, to resolve this case, always start by repeating the entire blood grouping procedure and verifying the patient history, particularly the age of the patient. So, just like on the previously discussed, newborns produces sufficient detectable antibodies between 3 to 6 months of age, while old age individuals or elderly individuals has a depressed antibody production. So, to resolve this case, always begin by requesting for a new sample and repeating the entire procedure. But if the result is still the same, incubate the patient's serum with your reagent A1 and B cells at room temperature for approximately 15 to 30 minutes. Okay, remember that ABO antibodies are IgM in nature, meaning they react best at room temperature, and prolonging the incubation time to 30 minutes will actually facilitate a more leeway time for the antigen and antibody to fully demonstrate bonding and agglutination or interaction. So, or can be this is a practical skills application. You can add 1 or 2 drops more of plasma or serum to the test. This is to enhance the antigen antibody binding between your reagent cells containing the antigens and your serum sample containing your antibody. Or by adding plasma, since also your plasma also contains your antibody, so it can be utilized to enhance the agglutination reaction. The main principle of this is to increase the likelihood of sensitizing more reagent cell antigenic determinants via addition of more antibody epitope sites in this test sample, which will increase more antigen antibody complex and a more visible agglutination. So, however, if there is still no reaction after the centrifugation, the test mixture or serum and cell mixtures can be incubated at 4 degrees Celsius for 15 minutes. However, when doing this class, you should always include an autocontrol and O cell control, since the lower temperature of testing will mainly cause your reactivity of other commonly cold occurring agglutinins such as your anti-I that has actually a high reactivity, most particularly to adult RBCs. So, okay, an overview. When we say autocontrol, this is mixing the patient's red cells and serum. This is used to detect if the person has autoantibodies or antibody to his own antigens. The positive reaction is the presence of agglutination in the autocontrol, and this is evident in cases of your autoimmune hemolytic anemia, which will be discussed properly in your hematology class. So this will be discussed on your hematology. And for your O cell control, this is a combination of your red cells of known blood group O samples and is used to rule out autoantibodies or alloantibodies. So in comparison with your group 1 discrepancy, group 2 discrepancies occur in the forward grouping and interferes with the detection of your ABO red cell antigens. So for the common causes of a group 2 discrepancy, we have presence of subgroups of A and B, leukemias like your chronic lymphocytic leukemia that yields hypogammaglobulinemia, the one we discussed earlier. One significant clinical correlation to your hypogammaglobulinemia is the non-Hodgkin's lymphoma that actually mimics the clinical signs of the antigenic suppression seen in leukemias. And lastly, your acquired B phenomenon, which was already explained into detail from our, of course, recent slides. And the primary resolutions are almost similar with that of the if your group 1 by incubating the test mixture at room temperature for up to 30 minutes. So the test mixture that we're referring to is the mixture of your red cells and the anti-sera, since we are dealing with forward grouping. So if the resulting reaction is still negative, incubate the sample mixture at 40 degrees Celsius for 15 minutes. While doing this procedure, never fail to include O cells and autocontrols, just like the group 1 discrepancy. This is to rule out the possibility of reacting with autoantibodies or alloantibodies. So this is a sample case on the common ABO discrepancy. Group 2 discrepancy, particularly on a patient with acquired B phenomenon. So remember that the forward and reverse grouping results always yields or demonstrates a very strong positive agglutination reaction of 3 plus to 4 plus. Also, given that reverse and forward grouping can be counterchecks with one's results, we can easily notice in this table that the probable blood type of the patient is blood type A. However, due to the presence of a weak agglutination reaction on the anti-B on the forward grouping, one can be confused with the reading as blood type AB. So, and that's exactly why we can determine the true blood group via counter-checking with the results on the reverse typing, which actually demonstrated a strong 4 plus agglutination on the B cells. So, a sample with a true anti-B reactivity on the forward must not have an agglutination on the B cells in the reverse typing. So, to resolve this, we normally acidify the anti-B reagent into pH of lower than 6 or can be raised to pH greater than 8. And why exactly? It is because a true acquired B antigen will not agglutinate on this mentioned pH values. Blood banks are already utilizing ES4, which is a readily prepared monoclonal antibody clone that agglutinate potential red cells with high concentrations of acquired B antigens. Okay, class, remember that in an acquired B phenomenon, the bacterial enzyme deacetylates the N-acetyl-galactosamine of the A antigen, modifying it into D-galactosamine that resembles your B antigen, which is actually responsible for the cross-reactivity to your anti-B in the forward grouping. This leads us to one of the resolution, which is the addition of acetic anhydride with the red cells. So the main action of this procedure is that it reacetylates the red cells antigenic structures, meaning to say it replaces the modified terminal sugar chains from the galactosamine into the original N-acetyl-galactosamine. And this action will result to the red cells with B pseudoantigens demonstrating a decreased reactivity, and the red cells with true B antigens will not get affected. So acquired B antigen can always be resolved using autocontrols and your direct antiglobulin test, or which will be discussed in a separate topic with presentation. The following are the rare causes of the group 2 discrepancy. The first one is due to an iatrogenic cause, like in conditions of the gastrointestinal related origins such as carcinoma of the stomach and pancreas. In this particular condition, this disease will initiate the production of high concentrations of the BGSS or blood group specific soluble substances, which may partially or completely neutralize the reagent anti-sera used for the ABO grouping. So it will leave no available binding sites for the anti-sera reagents since they were already occupied by the excessive amounts of the BGSS. So to resolve this discrepancy is to simply remove the bound BGSS by washing the red cells several times with normal saline solution in order to free the cells from the excess blood group specific soluble substances. So next rare case is antibodies to low incidence antigens in reagent anti-A or anti-B. Remember, class, that your ABO reagents were actually commercialized from both human and animal sources that has been screened efficiently, however, may not have been screened from low incidence antigens. Take note, class, that there is something that we call as private antigens. This is included in your Henrys, which is one carried by on the red cells of very few individuals. So when these sourced out reagents were utilized on the red cells of these people, will give an unexpected reaction of the patient's cells with anti-A or anti-B, or can be both, and exhibits a weak agglutination reaction, or mimics the presence of a weak reacting red cell antigen. So the resolution to this discrepancy is simply using anti-sera reagent with different lot number. And to conclude your list of rare group 2 discrepancies is your chimerism. You may probably have heard the term chimera before, most commonly from sci-fi movies, where there is a half human, half wolf, half human, half bat, and so on. Well, in blood banks, it is nearly similar to that case, and however, in a formal view, can be genetically identified as the presence of two cell populations in a single individual, which is predominantly seen in cases of twins having born to a group O mother and a group B father. Okay, these twins demonstrates two cell groups, which is both O groups and B groups, instead of the supposed B or O group only. The resulting pattern of agglutination is mixed field reaction. So this actually happens via gestational period, where both twins undergo utero exchange of blood via vascular anastomosis, which resulted to the merging of two cell populations. We actually categorize chimerism into two categories. We have your true chimerism and artificial chimerism. So the one previously explained builds the true chimerism, while for artificial chimerism, it can be caused by number one, out of group transfusion, which results when group O cells are transfused with a group A or B recipient. Okay, class, this can happen anytime when there's an emergency transfusion and that the patient is extremely critical of blood loss. Since your blood group O is the universal donor, it can readily be transfused with any of the blood groups. However, with given consent and a well-documented waiver from the immediate family. Okay, I just want to share your class that I recently had a patient which is a perfect example for this case. He is from a remote area in South Cotabato. He was stabbed straight through the heart, and his inferior vena cava was nearly detached from his heart. So it means that there was a very dramatic bleeding and extremely profuse blood loss. Of course, this case is directly sent to the operating room. But unfortunately, the hospital is deficient of his blood type, which is blood group B. But since his close friend is a provincial jail warden, this warden recruited and sent lines of prisoners at the blood bank to the hospital to suffice for a blood group O, bypassing now the close matching procedures. However, class, the patient contracted hepatitis B from the transfusion, but the patient was neither regretful since that gave him one more chance to live. This is one risk factor, however, of an extremely emergency transfusion situation where if you were located or the procedure was conducted in remote areas, having limited facilities and availability of blood samples or blood bag units. Since this came ages ago, and as of today, we have a more regulated and strictly mandated the protocols in the sufficiency blood unit storage, which were readily screened from infectious diseases. So moving on, bone marrow transplants or stem cells, these procedures will exhibit weak agglutination patterns from post operations and will eventually create or produce a different RBC populations during the recovery. It means therefore that the term artificial chimerism. So the number three is exchange transfusion, which almost has a similar concept with that of your stem cells. In this procedure, it is also so termed because the red cells of an individual are being replaced by another cell population that will eventually demonstrate two cell populations. And lastly, number four is fetal maternal bleeding. Next, we have group 3 discrepancies, which are caused by plasma dyscrasias or plasma abnormalities that results in rouleau formation or pseudoagglutination that interferes with your ABO grouping since it has in vitro occurrence or demonstration. When we say rouleau formation, these are stacks of red blood cells that exhibit a coin-like adherence pattern that mimics the appearance of an agglutination, thus the term pseudo-agglutination. So we have here the four main classifications of the causatives of the blood group 3 discrepancy. So the elevated levels of globulins in multiple myeloma, Waldenstrom's macroglobulinemia, and so on. Elevated fibrinogen levels, use of plasma expanders like dextrans, polyvinyl pyrrolidons as fluid replacement therapy, and a favorite board exam pointer, your Wharton's jelly, which is also termed as substantia gelatinea funiculi umbilicalis, which is primarily a surfactant made from mucopolysaccharides that serves as a cushion of gelatinous substance protecting the umbilical course. So take note that the main breakdown of this discrepancy is actually the presentation of pseudoagglutination that interferes with the ABO blood grouping. So here is a nice case presentation of your ABO discrepancy caused by rouleau formation. Remember class that when there's a strong agglutination of 4 plus in an ABO grouping that is clinically definitive of the resulting blood type already. So given the data of results, we can see that the probable blood type of this patient is blood type AB, given that it has a strong reaction on the forward typing for anti-A and anti-B, meaning to say we should expect for a negative agglutination on the reverse typing. However, in this case, as you can notice class in this table, it shows a weak 2 plus agglutination reaction, which can be deduced that it may be resulted from pseudoagglutination caused by rouleau formation due to an increase in serum or plasma protein concentrations. And to resolve this discrepancy, we can perform a microscopic examination and search for the evidence of the rouleau formation, and also saline replacement technique, which wherein we replace the serum with saline of equal amount, and the action of this procedure will enable us to determine a true agglutination. Okay, why? Because the saline replacement technique will not affect the true agglutination, while pseudo-agglutination will demonstrate weakening of the reactivity. And the same principle applies with washing cells 3 times with saline solutions, while in the case of Wharton's jelly, 6 to 8 times of saline washing will slowly alleviate the pseudo-agglutination. So while washing the red cells with normal saline solution, it will provide sufficient resolution for the Wharton's jelly. And lastly is to run auto what you call that an antibody screening or antibody panel. So that will be discussed on separate week discussion. So unlike the first three groups, your ABO group 4 discrepancies are basically due to miscellaneous causes, which includes cold reactive autoantibodies, or when the patient has circulating RBCs of more than one ABO group, unexpected ABO isoagglutinin or antibody, and unexpected non-ABO alloantibodies. And lastly, the Cis-AB phenotype, which is characterized by the inheritance of both AB genes from one parent carried on one chromosome and an O gene inherited from the other parent, having a very rare case of a progeny having the three ABO genes instead of two. So cases were first recorded from a Polish family in 1954. So as for cold reactive antibodies, we have learned that most of the ABO antibodies are IgM and that are cold reactive. And since we are talking about cold reactive antibodies, we are referring to the serum typing. So basically, in short terms, cold reactive autoantibodies is demonstrated when RBCs are so heavily coated with the antibody that they spontaneously agglutinate. And take note that they are independent of the specificity of the reagent antibody. So one most common example is your anti-I, which is present predominantly in adult RBCs. This antibody can exhibit pan-agglutination, which gives this antibody the ability to agglutinate all of the normal cells in a particular population. So while with patients that have circulating RBCs of more than one ABO group, maybe due to RBC transfusion or marrow stem cell transplants and out of blood group transfusion, which was already discussed and mentioned previously. So for unexpected ABO isoagglutinin, one perfect example is your A1 and A2 blood subgroups of A, which I have discussed in the previous slides presentation. And lastly, there are certain individuals who may have antibodies against your yellow dye used most particularly with your anti-B reagent. So the obtained data of results in this present slide shows a noticeable probability of having blood group B, since anti-B in the forward typing demonstrates a strong 4 plus agglutination and 4 plus agglutination on the A1 cells in the reverse typing, which we can correlate with the inverse reciprocity or correlation of your both of the ABO blood grouping. So, however, on the reverse typing, B cells shows a 3 plus agglutination, but whenever there is a strong 4 plus strength of agglutination, it is always regarded as the most definitive basis in ABO grouping. So, in this case, therefore, this strong 3 plus reaction demonstrated in your reverse may be caused by the potent anti-I, while the weak agglutination observed in the anti-A of the forward grouping is caused by your consequent agglutination of the antibody-coated red cells. So to resolve this discrepancy, incubate RBCs or serum at 37 degrees centigrade in a short period of time, and then washed with warm saline at 37 degrees Celsius or centigrade temperature 3 times and retype accordingly for both forward and reverse. So in forward typing, when the washing did not correct the agglutination reaction, the addition of a zero point 0.1 to 0.01 molar of dithiotretol is necessary to disperse the cold antibody related to the false agglutination or the cross reactivity of the cold antibody, which is anti-I. So in the serum typing, the serum red cell samples are actually incubated to warm at 37 degrees centigrade and then mixed and then washed with warm saline solution and retyped accordingly. However, if the same reactions observed are still unchanged in the reverse typing, or for example, when there should be an expected agglutination reaction, that is actually we utilize the cold auto-absorption procedures, where we utilize the patient's cells and made them to react with his own serum in order to remove the cold antibody from the serum. And lastly, run direct antiglobulin test, an autocontrol, and perform an antibody screen. So these three are complex topics which will be discussed in separate week discussions. So this is a recall and a review from your blood group A subgroup. We have learned that from our earlier discussion with the basis of the genetics and the concept of the A to B subgroup structural formation, that individuals of this group actually accounts for 22 to 35% of the likelihood of producing anti-A1. Your anti-A1 uh, isoagglutinin from your A2 subgroup, which is evidently observed as a weak reaction on the A1 cells. So as you can notice that the main ABO detection hindrance here is the weak reaction on A1 cells in the reverse grouping. And to resolve this discrepancy, we can use the dilocos bifluoroselectin, which will agglutinate only A1 but not A2 subgroups, which also includes serum testing with A1, A2, and O cells. And an autocontrol will give a sufficient resolution procedures for these unexpected ABO isoagglutinins. So for our last ABO case presentation is the discrepancy caused by unexpected non-allo antibody. So as you can notice that anti-A and anti-B shows a strong reaction in the forward grouping, which means that it is a probable blood group AB. And the expected reverse of the serum antibody typing results must show absence of the agglutination reactions. However, in this particular result data, there is a demonstration of a weak reaction on both A1 and B cells in the reverse typing, which is most likely to be considered simply as weak reacting alloantibodies. So the best resolution for this case is the performance of an antibody screen and panel. And okay, as I keep on reiterating multiple times, this will be uh, discussed in a further detailed manner on separate presentations. So that ends our discussion for the ABO blood group system, and I hope everyone made good notes during the discussion. Most importantly, I hope everyone have understood the concepts in the ABO blood grouping. So thank you for your time and diligence. As an advantage of having a pre-recorded mode of delivery, you can always revisit and rewatch the entire or whichever topic you interest more of understanding. And if you have any questions or something you're curious about that are related or that is related to the main discussion, feel free to message me on these accounts. So once again, thank you and have a nice day.